Hi, today is Unit 1, Lesson 5. It's our second day of Lesson 5 about our model ecosystems, and today is the first day we will actually see our model ecosystems. So you'll need the following things in class. You'll need headphones, your computer, a pencil, a dry erase marker, and your science binder. That's what it says behind my pictures here. And on the whiteboards in the classroom, I'm asking everybody to write the answers to or respond to these three prompts. So for part A, it says, what are 3 factors that can affect climate? You can pause the video and think about your answer and then turn the video back on to see if you're correct. So we talked about 6 factors. We talked about sunlight, oceans, winds, land types, clouds, and human activities. Those are all things that can affect climate. Name a physical adaptation that a plant might have. So you have to think about plants and some of the adaptations that we have talked about. And you might be thinking of spiky leaves, or spiky needles is actually what we normally would call them on a cactus, for instance, or even on a pine tree. Or some plants have a waxy coating. Some of them, like poison ivy, can produce a poison that causes a rash. So our plants have lots of physical adaptations. And finally, list a qualitative observation about the classroom. Lots of things that you can think of in the classroom, and you might say that the walls are blue. The temperature is usually warm in our classroom. The teacher is audible, which means we can hear her, because your teacher is often pretty audible. All right, so since there are dry erase boards, large dry erase boards on your desks now, I want to talk about using dry erase markers. You should have your own dry erase markers with you, and you should have your own pencil and that sort of thing. But today I do have some dry erase markers out on the tables. There are 4 on each table. Hopefully we will still have 4 on each table at the end of class. So take a look at this dry erase marker. The felt tip right here, that's our writing part of it, right? That's actually, the tip of the marker is called the nib. And it's kind of a felt-like material that releases ink when you press it on the whiteboard or when you wipe it across the whiteboard. It does not need pressure. So please be gentle with the dry erase markers. If you press too hard on the nib, you will ruin the marker. Please don't do that. Always replace the cap when you're not using it so that it helps that marker to last longer. And at the end of class, be sure your board is wiped clean. Please always be gentle with the board. These boards have lasted a couple of years and we would like to have them last for longer. And finally, dry erase erasers are used to wipe off the boards. Please do not write on them, do not pull them apart, do not lick them, do not chew on them. Just put them gently back in the baskets. Pretty please, be nice to the boards, the markers, and the erasers. All right, so down to what we're going to do today. We're going to learn about how to properly use tools to measure water quality. We are going to look at the quality of our water using certain tools. That's because we want to accurately describe and observe the model ecosystems to determine how healthy they are. So we need them to be healthy if we have living things in them. So what type of model did we say we were going to create and observe in class? We said it would be a model aquatic ecosystem. And eventually this is what our ecosystem will look like, and then we will have more things in it too. But today, it actually doesn't even have plants. Today, this is what our model ecosystems look like. So, I'll back up. So we want to see how the changes in the abiotic and biotic factors, so the non-living and the living factors, have an effect on that ecosystem, on everything that's in the ecosystem. All right. So are they ecosystems yet? Not yet. Ecosystems have to have both biotic and abiotic parts or factors. And right now, this one only has abiotic factors. We have water and we have some rocks and some sand. Safety first. Today we will be using our testing equipment, so we're going to need goggles. So you should find the big baggie for your table in the middle of your table, and it will have your goggles. If it's not there yet, your teacher will be putting it there shortly. All right. Also, everybody's getting their model ecosystem packet today. You need to keep track of this packet. It does have 3 holes punched in it so that it can live in your binder. You need to bring it back and forth to class each time, and we're going to be using this for about 4 weeks. When you first get this packet, please put your first name and your last name and your class period right on there. Don't worry about the bottom. We're going to talk about the bottom in a second. First name and last name, not just your last initial. Now, at your table, I want you to decide, there are 4 people probably at your table, decide what letter you want, A, B, C, or D. Each one of you gets 1 letter. So there can only be 1 A, there can only be 1 B, there can only be 1 C, there can only be 1 D. If you only have 3 people at your table, each one of you will still choose one of these letters, and then each day we will see who is available to do the 4th test, and you'll understand that a little bit more once you see what I mean by testing. All right, so once you've decided what letter you are, put your letter down on your front of your packet, and your group number is just your table number, whatever table number you are at in your classroom. 1, 2, 3, 4, 5, 6, 7, or 8. Those are your options, and it should be written down on your baggie with your goggles. All right, so don't lose this. You're going to be turning in it at the end, but you'll need it for 4 weeks because we're going to take data often. So which abiotic or non-living factors will you be observing and recording about your ecosystem? Look here, this is our data table. We're going to record the day, we're going to record the date. This is temp or temperature. DO is dissolved oxygen. pH is a measure of acidity. Nitrite is a component that we could find in the water. We're going to measure water clarity as well. Also, we're going to make some qualitative observations, and we're going to make some inferences. So the important things that we are thinking about right now that are the abiotic factors, that's our temperature, DO, pH, nitrite, water clarity. And so these are going to be quantitative when we are talking about temperature, dissolved oxygen, pH, nitrite, water clarity. Qualitative observations are just going to be qualitative, and inferences are going to be a combination. So mainly looking at what we are testing in class, it's going to be quantitative. All right, we'll be testing for temperature. You do know temperature already. How hot is it? How cold is it? So depending on the day, you might be using a probe to place inside your ecosystem to check the temperature, or you might be using an old-fashioned thermometer. So we have both of those, and it just kind of depends on the day which one you'll be using. Dissolved oxygen, you're going to learn more about today while you're taking your notes in class, but it's the amount of oxygen that has been dissolved in the water, and we have a special probe to measure that. So the probe looks like this, and if you look at the very bottom of the probe, it has a sensitive covering that can help calculate the amount of, or measure the amount of dissolved oxygen in our ecosystem. We're going to measure pH. pH measures the acidity of our ecosystem. So the pH probe looks a lot like this one. Please be careful on the pH probe. And then I'll tell you about this, sorry. The pH probe is super, super fragile. So just assume all the probes are super fragile. But this one, if it breaks, we will not get another one. So just be super careful, please. That is glass, and it really does break. Gentle, gentle. So pH is actually written with a lowercase p and an uppercase H. You don't have to know this, but it stands for potential hydrogen. So that's going to tell us whether it's acidic or not. All right, we use a device that can read those probes and give us our numbers digitally. So for this particular device, it's reading the pH value as 3.93. You can see that pretty clearly in the picture. This is actually dissolved oxygen, or DO. So when you're looking at the device, you'll be able to see those letters better, and you can see the numbers for dissolved oxygen. And then in this case, these students were able to measure temperature as well, and it was 21.7 degrees Celsius. So sometimes you'll see all three of those numbers on your device, and sometimes you'll actually only see the top two. That will change from day to day. All right, and then the next thing that we're going to be testing is nitrites. So nitrites are chemicals that we find in the water, and we're going to test for the number of, or the amount of nitrites that we have in our ecosystems. And then we're going to test for water clarity. So it's kind of like it sounds. How clear is the water? This water here is very clear, but on the right side, that is pretty murky water. You can't see very deep into that water. And so we have a scale that we're going to use. Whoopsie daisy, excuse me, let me come back. So if it's really pretty clear, we're going to give it a number of 1. Clear is that number 1. 2, it's still reasonably clear. And then as you get murkier and murkier, we get to our value of 5, which is pretty murky, pretty hard to see through. And this card that has these numbers and these examples, that will be on each one of the ecosystems, so you can check it. All right, so right now I'd like for you to find this page in your packet. It's pretty much the first page after the title page, and you are going to be drawing your ecosystem. This entire box is your ecosystem, so I want you to pretend like this box is your ecosystem when you're drawing, okay? Don't make something small in the middle. I want you to use this whole thing is your ecosystem. Okay, so this whole thing here is your ecosystem. It's going to fill up your whole box. Right now, you can use a pencil and you can kind of sketch across the level of the water that you can see, and you can sketch roughly where the gravel is, and in each one of your ecosystems you should have a bigger rock as well. So just have an idea of what your ecosystem looks like. All right, so these are the things that we're going to be measuring: temperature, water clarity, dissolved oxygen, pH, and nitrites. So I'll ask in class if anyone happens to know much about these, and then we're going to be learning about them in class today, so I won't say anything on our video. So we're going to learn about each one of these abiotic factors. We're going to learn how to test for them, and then we are going to practice how to do each one of those tests. All right, there are links in today's Schoology folder, and you're going to use the links in the Schoology folder. There are files on those links to fill in all of these boxes. Please don't be overwhelmed. You're going to do one section at a time, okay? And I'm going to help you figure out which section you're going to do. So you're just going to do one section, then you'll do another section, then you'll do a third section, and then you'll do a fourth section, just one at a time. So each one of these is a separate little section in your packet. All right, then, so on this page, on these pages, you're actually learning about each one of these elements that we're talking about. And then we have a page where you're going to practice. You're going to go up to your ecosystem and you are going to do some testing, and you're going to record some numbers, some quantitative data. Okay, so you can see that we're going to measure how warm the temperature is in the water, and we are going to find a number that represents dissolved oxygen in our water. We're going to record the pH, which means how acidic that water is, and we are going to take a look at whether there are nitrites in our water. Nitrites are actually harmful, so we don't really want there to be nitrites. How clear is our water? We don't have much in there, so we hope it's pretty clear today, but you'll see. And then, are there some qualities you can describe about your ecosystem? Water color, color of the rocks and things that are in there. Are there any new organisms? And can you describe the things that are in your ecosystem? All right, so you'll need to have goggles on once we get started because people will be testing. So when you have your goggles on, you're going to be going through answering questions on those pages in your packet. And since you're using slides that are linked in Schoology, one of the slides is going to say, now take your packet back to your ecosystem and see if you can test your particular water quality. Okay, be careful moving around the room. There are a lot of people in the room. Now there are ecosystems in the room. And so hopefully your backpacks are all tucked underneath the tables. All right, so think back. What letter are you? What letter did you write on the front of your packet? If you are letter B, I'm going to say that I'm letter B. The first information I'm going to read about and write about is dissolved oxygen. Okay, so that's where I'm going to start because I'm letter B. Then, after I finish my dissolved oxygen, I'm going to do pH. I'll finish all of pH. I'll do nitrates, finish that, and then I get to do temperature and clarity. So this is helpful because we will be able to spread out around the room a little bit and not be on top of each other. All right. And then, depending on how much time we have in class, we are going to learn a little bit about how scientists actually find about water quality in the real world. So the quality of our waterways is constantly being monitored by scientists. This is a scientist who's out in the field getting ready to do some of the same exact tests that you have practiced today. It looks like this. Scientists are out oftentimes in streams and right at the edge of ponds or maybe even on boats taking water samples. And so we have a video that we can watch in class that I'm not going to record here, but I can share that video if you miss class and let me know. I will share the video with you. Please check in Schoology to see if we had time in class to get to our exit ticket. If there is an exit ticket called U1L5 Day 2 Water Quality, you'll want to be sure you do that. Or just check and see if you have any assignments that are going to be due shortly. Thank you so much for watching.